Hello and good morning, everyone. My name is Masoud, and today we're going to talk about mathematical modeling. So we will try to cover the basics of mathematical modeling, what they can do and what they cannot do, and get a little bit familiar with different types of mathematical modeling that are available, and basically learn how to build and analyze a mathematical model similar to an experimental study, as we will see. So the first question is, what is a mathematical model? Or more general, what is a model? Well, a model can be an animal. We use an animal to basically understand the behavior of the bigger population or even different species. For example, we study mice to understand how the immunity works in the humans or in mammals. A model can be a prototype, like this little building. We use it to understand the properties of a bigger system. Or it can be a mathematical equation. And mathematical modeling is basically when we change or when we try to describe a system with a mathematical equation. So in all of the models, basically when we build a model, we try to understand how the system works. And that means we need to do some level of simplification in our model. But why do we need models? Well, basically, models provide us a tool to understand the system and see how different elements of the system interact and work. It helps us to predict the outcome of the system, and that is very important because we can make policies and we can put interventions to amplify or to avoid something to happen. For example, all the interventions that were placed for COVID, I hope, were based on model. And also, it can help us to predict the outcome of those interventions. But why mathematics? Well, because it's awesome, really. It has been there since the day one. We learned how to count, and it has evolved in such a strong language that is so precise. There's only one way you can understand, one way you can interpret a mathematical equation, not two opposing ways. It is very concise. You can have one equation that explains thousands of words to you. And it's universal. It has been used in many, many different topics and subjects. And finally, and I think the most important, is that it's best understood by computers. It's the best language that computers can understand. So another benefit of mathematical modeling is that we can synthesize and put together our fragmented understanding of a system. We do an experimental study on a very narrow subject, and we get a little bit of understanding for that particular subject, similar to seeing a pixel in a bigger picture. Our experimental studies are like that. Mathematical modeling helps us to connect these pixels together and see the bigger picture. I've shown you here a pixel of a bigger picture. Can you guess what it is? Okay, I mean, it's not fair. It's only one. Maybe a few more. Sheep? Yeah, very close, but not close. Not yet? Yes, very good. It's an elephant. So this is, yeah, I know, it was just really small. So similar to this, we get understanding from different experimental study by combining them with mathematical modeling. It can also help us to find out what we cannot measure. For example, tomorrow we'll see that for resilience, there is not really something that we can go and measure in the farm. Resilience, you have to have a mathematical modeling to measure the resiliency of our animals. Again, I'm showing you an example. Can you guess what this is? Again, an elephant. And finally, mathematical modeling does not have the constraint that we get in real world and by physical constraint or by time constraint. For example, in the context of infectious disease, we cannot basically have the resources or the time, or it's not ethical to infect half of the population and see how the epidemiology grows. And there are like many what-if scenarios that we do not basically have time and resources to do that. But we can simply do that with mathematical modeling. So in summary, it helps us to combine our fragmented understanding of the system, and that helps us to have a better understanding of how the elements of the system interact with each other and how the system works as a whole. It can help us to test hypotheses and make predictions, which is also very important in doing risk analysis and in making policies. But mathematical modeling also comes with their own limitations. The first one is the lack of quantitative knowledge. Well, imagine you want to model the host parasite inside a sheep. You need to know the biology of the sheep. You need to know how the immune system of the sheep works. You need to probably know about the life cycle of the parasite. So there are many important information that you need to know before you start doing this modeling. And that may not be available. You might need some data to start your modeling. Or you might not have the right tools to estimate a parameter. For example, in the context of epidemiology, measuring susceptibility and infectivity is quite difficult. Well, recently there has been studies that can facilitate this kind of measurements, but there are still many traits that are very difficult to estimate. And finally, biological processes have an inherent randomness in them. And with mathematical modeling, we cannot for certain predict the outcome. For example, we cannot say for sure if an animal will get infected or not. We can provide probabilities, but like bringing a definite answer is something we cannot do. So now we have learned a little about mathematical modeling in general. Let's see what types are out there. So depending on the criteria you use, you can divide mathematical modeling in different groups. We have empirical versus mechanical. We'll talk about that in the next slide. We can have deterministic and stochastic. We'll talk about that too. Depending on the scale you use, you can divide your mathematical modeling all the way from systems modeling down to very small, like the scale of molecules, or even things in between. Static modeling refers to when we want to basically understand the interaction of variables at one instance of the time. If you are also interested in the interaction of the variables over time and also like how variables interact with their past, then this is called dynamic modeling. That means we have the time element in our model. Depending on the function you use, it can be called linear or nonlinear. So there is a very famous sentence in mathematics that we say, dividing problems into linear and nonlinear is like dividing the objects in the world into bananas and non-bananas. So there is like hardly ever any mathematical problem or any real-world problem that you can just use linear function and be happy. Well, in certain cases that you are like looking at a very narrow range, linear functions work, but in general, everything is nonlinear. And then depending on the variable you use, it can be divided into discrete or continuous. For example, number of infected animals is a discrete variable, whereas time, body weight or temperature is a continuous variable. Okay, so empirical models are basically when we have some data and we want to understand what is the relationship between these data. So our starting point is our measurements that we take in the field. For example, we have a milk yield and days in the milk, and we want to understand the relationship between them. We don't need to know biology. A simple regression will give us the answer. So there is no prior knowledge about the biology of the system. On the other hand, we have mechanistical model. Here we need to know the biology, and we want to test a hypothesis that we have observed in some other data. For example, we want to know how the pathogen affects the body weight, the body weight of the individual, or the growth rate. So this is our hypothesis. We know that infection should affect the body weight of the individuals. So the aim is to understand how it works. So we make a mechanistical model, but in this case, we need to know the biology of the system. We need to know, for example, the life cycle of the pathogen, the nutrition and the energy allocation for the animal. But we don't need to start with the data, but we need the data at the end to calibrate our model, or basically to validate whether this model actually works in real data or not. Deterministic versus stochastic, this one is probably you already can tell by just the name. So if you always get one output with a given input, then your model is deterministic. So for one input, you always get the same output. However, if for one input you get an ensemble of outputs, because there is randomness incorporated into the model, then this can be called stochastic models. Usually you have a combination of both. For example, the effect of a nutrient on the growth. This is deterministic. One nutrient will always have the same energy, but like the breeding values of the animal will be a random variable. So if you have a system where you want to look at the effect of nutrition on the growth, then this is a hybrid model where you're using both. So this is the last slide on the type of the mathematical modeling before we start looking at how to build models. Mathematical modeling can be divided into the groups, different groups based on the model you use. So for example, if you are selecting the best animals in the country based on a trait, you're basically modeling at the national level. If you are looking at what affects the growth of rumen in dairy cows, then you are looking at an organ level. Or if you are looking at the gene expression based on a nutrient, then you are looking at the molecule and gene level. So normally what you do in reality, you always combine like one or two or three of these scales. For example, in the afternoon, I think we will see the modeling of milk yield for animals. So that's at the individual level. If you try to select which animal is better in terms of milk yield, then you're also at the herd level. If you combine many of these scales, then these are called system models. Before we continue, I'll just tell a little about another word that comes up, simulation model. This is not a different type of mathematical model. This is basically when you take your mathematical model and put it inside a computer, you implement it, that's what we call, and you run your simulation, which means that for a given input, you get a set of outputs. Okay, so now we're going to see how we can build models, or how we can do modeling. So modeling has 4 stages and is very similar to an experimental study. In an experimental study, you start with a question that you want to answer. You design your experiment to answering that question. Then you do your experiment, you collect your data, you analyze your data, you validate your findings. If they make sense and they are basically what you expect, then you're happy and you go apply it in the field or in practice. If not, then you redo your design and redo your analysis until you get to the answer. In mathematical modeling, we do the same. We start by building a model, then we generate prediction and analyze our model, which you could do it in a computer, basically generate prediction by simulating. And then you validate your answers, basically your predictions, if they make sense and if they are in line with your expectations and literature, then good. You apply it to the real data or you release your model. If not, then you redo everything. So now we are going to look at each of these elements in a little bit more detail. So similar to an experimental study where we start with a question and we want to answer, the first step of building model is defining your objective. It is really important. I think it is the most important step in mathematical modeling. Is it the milk yield of the cows you want to predict? Is it the effect of a gene on certain trait you want to assess? Like, define your objective and be very clear about it. Then comes the question of the appropriate level of difficulty or simplifications you need to do in your model. For example, we want to model the effect, we want to select animals that are more resilient to a certain disease. Do we also take into account the life cycle of pathogen? If you're talking about virus, do we take into account the different frequency of different strains of the virus, or do we not care? Because whatever you do, you're never going to exactly imitate reality. As George Box says, all models are wrong, but some are really useful because they give you predictions that are very close to reality. So in that sense, if you have two models and they give you the same prediction, we're going to favor the one that is simpler, that has less parameter, and we will see why this is a very good approach. And this has been said by William Ockham. No, so now you have a level of simplifications built in to your model. The next thing you need to do is make assumptions. And the making assumptions is very important because whatever assumption you make is going to directly affect your result and your findings. This is very clear with the physics because Newton said mass is constant, and he developed all the classical mechanics. And then Einstein said, oh, mass and energy added together are constant. And then he developed a completely new field of physics. And there is no way you can go from Newton assumptions to relativity theory because the assumptions are different, so the findings are going to be very different. Let's look at an example, actually, a detailed example. So we say a population growth is proportional to the size of population. That means if I have a population of 100 animals, and if I have a population of 200 animals, the one with 200 is going to grow twice as fast the other. So if I say that, then I can write my model this way. So that means that P here is the population size. dP over dt, which is the growth rate of the population, is equal to P multiplied by A. So the growth of the population is equal to the size of population times A, which is a constant. So this is a very simple model. You can solve it analytically to get to the formula, Pt equal this. So that means that population size at time t is equal to the population at time 0 times this exponential function. Now I made my model, I'm happy. But actually what I'm assuming here are very important assumptions. First, I'm assuming that there is no limit to the growth of the population. It can go infinitely large. The second assumption is that growth is a continuous process. So I can have 14.65 individuals. I cannot have this I can have any individual with any number, like not whole numbers. The other one is we don't make any assumption on P0. So basically, I'm assuming that you put one individual here, and after 5 days, there will be 12.35 individuals. And then I'm not modeling the death. So I'm assuming that all the individuals live forever. So if any of these assumptions are wrong, which they are, then your model is wrong. Another thing that can be really useful in modeling is to have a flow diagram about your model. So flow diagrams basically tell you the state where your variables are changing or how your variables interact with each other. For example, in the epidemiology model, we know that we have a group of susceptible animals that become infected, and they eventually are recovered or dead, removed. So the arrows go from susceptible to infected and then to recovered. But then infected animals can become infectious and affect the susceptible population. So there is a feedback going from infected to susceptible. This was a simple system, but like in complex system where we are considering the life cycle of virus and also the pathogen and the host, this becomes really clear because now we're combining, I don't know, 2 pages of mathematical equation into one simple diagram that tells us how the variables interact with each other and how the states and how the system works. And finally, when you do that, you're ready to write your model equations. So if you're using a statistical model, you probably are going to end up with linear functions or nonlinear functions. If you're deterministic models, as you will see in the afternoon, right after this, I think, in Nick's talk, we will use differential equations. And if you're dealing with the stochastic models, you have to use probability distributions and so on. But whatever you do, please use something that already exists in the literature. If you find yourself in a situation where what you're doing, there is nothing even remotely close to what you're doing, then you probably didn't look at the literature correctly. Because there is always something similar to what you have done. Maybe not exactly in the same field, but something closer. For example, the Wood model that is used for lactation care can actually be applied to the within-host infection. And you can now use all the findings of the Wood model in that context. Apply it. OK. So now that you have written your equation, you can solve it. You can do that either analytically, if your model is very simple. For example, in the population growth, we had a very simple model that we could just solve analytically. But as I said earlier, this only works where you have linear equations and where your equations are very simple. And as I said earlier, the nonlinear and linear problems are like non-bananas and bananas. And you probably are going to end up at the situation where you cannot solve it analytically. And there you would need to use numerical solvers. Again, if you find yourself in a very complex, with a very complex model where you have to implement a numerical solver, do not start writing your numerical solver. Do not reinvent the wheel, because there are literally tons of softwares, free or paid, that can do the job. They have been tested, and you're not going to be able to beat them. So just use them and save yourself a lot of time. OK, so what are we now? We have built our model, and we ended up with a mathematical equation. We solved it, and now we want to start applying it. But as you probably have seen in simulation studies, you can easily be overwhelmed with hundreds of scenarios and cases that you want to consider. And before you even know it, you have a lot of scenarios to consider. But which scenarios you should really do? So there's a simple answer here. Just focus on the question you wanted to answer. If you wanted to answer the prediction of milk yields in second lactation of the cows, you don't need to care about, I don't know, the case for goat, or the case for first lactation, or the case for all the breeds of the cow. You just focus on the question you want to answer yourself. And also, the cases that you want to simulate needs to be realistic as well. But also, you want to extend your model a little bit just to look at the boundaries, what happened. So for example, if you're modeling an infection, you want to look at the boundary. What happens if I have only one infected individual? How the epidemiology is going to grow with only one infected individual? Just to see the basically the limitation of your model. And when you're using a mechanistical model, you have a lot of parameters to tune normally, because these models are complicated. And for them, you have two options. So you can either use the literature values for those parameters, or you can estimate them using many of the statistical methods that are out there. And now here comes the thing I told you about simple model. If you have a simple model, and if you have a complicated model, and they both give you the same output, then when it comes to the task of calibrating your model, the simple model, for example, like the population growth, I had only one variable to tune. And I can do it with only one least square. But if you have like 50 parameters to tune, then your job is going to be really difficult. So this is where the Occam principle makes sense. The simpler model is better to use. OK, so again, a wrap up. What have we done so far? We have built a model. We have made an equation from our model. We have solved our model. We have decided on the cases and scenarios we want to look at. We have tuned our parameters. But for example, we wanted to study resilience. But there is no data in real life where you can compare your resilience to the data in the literature. But probably you simulated also body weight. So what you can do is you can look at the body weight. Remember the elephant? The elephant is resilience that we cannot really measure, but we have the body weight. So we can look at the prediction of our model on the body weight, because these are like the intermediate variables that our model produces. And then we compare the body weight with the real data. And then with this way, we can see if our model makes any sense. And finally, we are ready now to produce prediction from our model and to see if they make any sense. Our model can now be analyzed and see, for example, I have 5 scenarios, and I get 5 outputs. The outputs are not very diverse. So I need to look and see what is happening, why my model with 5 different scenarios or 5 different outputs is not giving me 5 different inputs, is not giving me 5 different outputs. What is happening? What is the mechanism that doesn't allow this variation also in the output? Or on the other hand, I have 2 scenarios that are very similar, or I get completely different outputs. So there are techniques that you can apply to understand what is happening with your model. This is going to be a little bit technical, but I try to simplify it. The first thing you want to look at, if you are dealing with a mechanistical model, is asymptotic behavior. So you want to look at your model and see what happens in infinity. For example, if you are looking at infection, you want to see what's happening after, say, 10 years. If, for example, after 10 years you have 5% of the population who will be always infected, then your model has a steady state. And your asymptotic analysis tell you that at infinity, which is here at 10 years, you will always have 5% of the population who are going to be infected. But in some models, you will have no steady state. So there will be, for example, a seasonal infection that comes and goes. So here you will not have a steady state. Another thing you can do is initial phase behavior. So here, basically, you analyze whether your system is stable or not. In the context of, again, infection, you understood that 5% of your population after 10 years are going to be infected. Now, what if initially, instead of, I don't know, 20% infected, I have 30%? Am I going to still get 5% of the population infected after 10 years? If so, your system is stable. There are many systems in biology that are stable or unstable. For example, the sugar blood in your body for a person that has no diabetic is stable, is always a constant level. When you eat a meal, it suddenly goes up, but then your pancreas comes to the rescue and your sugar blood comes down, and after about 2 hours, your sugar blood comes again constant. So this is a stable system. For a person with diabetic, it's not unstable, but it just takes a longer time to reach. So this is basically where you can see whether your system is stable or unstable. And there are mathematical ways where you can actually do this properly and with rigorously and see and determine the stability of your system. The other techniques you can do are uncertainty analysis and sensitivity analysis. These are often confused. So Uncertainty analysis is basically you have one input and you want to see how certain or how confident you are for the output. Whereas the sensitivity analysis is kind of the reverse. You want to see how changes in the input affects the output. And they are very important. This is one of the key important aspects you want to do in analysis of your model. Again, I'll refer back to the example of population growth. There we had only one parameter, and we could change the constant A and see how it affects the growth of the population. If you have 10 parameters, then you have 10 degrees of freedom, or and like 5 inputs, so you have 15 degrees of freedom. And this becomes really difficult. There are sophisticated approaches like Latin hypercube sampling, but other methods that you can do, and it's simpler, is you keep everything constant. You just change one input or one parameter and see the effect on the output. OK, so now the model is built. We wrote an equation from the model. We solved it. We decided on a set of scenarios we want to check. And we got the analysis done, and we're happy with our model. Here comes the third stage, which is validating our model. So you have an experiment done. You get an output, observed output, and you predict based on your model, and you get a prediction. And basically, you can use standard summary statistics to see if your prediction and your observed variable are close. And you see that they are not. Well, the observed variable is far from the prediction. So basically, my model is garbage. Well, maybe not. That was one experiment, and you did one prediction from your model. If you make multiple predictions, many predictions with very similar inputs, you get a confidence interval for your output. And if your observed value lies in that confidence interval, then you're good. If not, you're probably in trouble. Your model probably misrepresents the reality. It doesn't have enough processes involved in it, or you're using the wrong equation, or your assumptions are incorrect. So you need to start all over and redo everything until you get something that is close to the observed variable. Finally, it becomes the case where you have multiple models. You have made multiple models. All of them give more or less the same answer for prediction. Which one do you choose? So here again, if you have independent models, you can apply the Occam's-Razor principle. You favor the one that is simpler, but also you want good predictability. I think in the lecture after this, Nick is going to show us a model where we look at the milk yield, and there we have a penalizer term, because it is very important that your model is not too flexible. We want predictability, we want accuracy, but we want also to make sure that the model is not too predictable and not too flexible. So there's a penalizer term there that it basically doesn't allow the model to overfit. If you have nested models, then it's easier. You use AIC or BIC to compare models and see which one performs better. And finally, you are ready to apply your model into the real case and do many interesting stuff from it. So you can apply this for risk assessment or to predict outcomes and make policies. And when you basically release your model, you have to be very transparent of the steps you have done, and you need to always remember that this model was built for a purpose. So have the user case in mind always when you do your model. So that brings me to my last slide, which was quite fast. So in general, a good model, as put by Einstein in his word, is as simple as possible, but not simpler. Your model needs to be fit for purpose. You want your model to have a good prediction and good accuracy, but you want also not to overfit, because overfitting is a big problem. it will give you nonsense result. And it is made for purpose. It is for a very, you know, a special case. You cannot simply take someone else's model and apply it in your own data without considering the user case. There is no free lunch. You cannot get something by doing nothing. You have to follow this procedure. And finally, if you are doing a predictive model, you need to parameterize it from the data available. And always remember that whatever you do, your model is not going to be perfect. It is not going to exactly represent the reality. But if it predicts good, then your model is good enough. So all models are wrong, but some are useful. And these are some interesting references that if you're interested, you can have a look. So that brings me to the end of my talk, and it was very quick. Thank you very much. If you have any questions.